Welcome to Microlab Mastery. Today's topic, negative stains. Negative stains are easy, quick, and do not require heat fixing, so the bacteria don't shrink. Slides should be handled with care because the microorganisms are not killed. Negative staining allows you to view the size and shape of bacteria cells, but does not allow you to differentiate between bacteria. Negative staining is useful to view bacteria that are difficult to stain or resist basic stains, such as mycobacteria. This process uses acidic stains, such as nicrosine or India ink, which are repelled by the bacteria's surface. The background is stained and the bacteria remain unstained. The material you will need in order to perform the negative staining will be bacteria cultures, microscope slides, the negative stains, a staining tray, a microscope, plus lens cleaner, lens paper, and immersion oil, and materials for aseptic technique, for example, an inoculating loop and Bunsen burner. In order to begin the negative staining procedure, first label your slide using a Sharpie. Next, please place a single drop of acidic stain at one end of your slide. Next, please follow aseptic technique to sterilize your inoculating loop, as you have previously learned in the class and as is being demonstrated here. Next, following aseptic technique, please flame the mouth of your test tube, and then, when your loop has been cooled, aseptically transfer bacteria from the culture to the acidic stain on the slide. When you transfer the bacteria into the stain, ensure you gently mix the two together. Then, please re-sterilize your wire loop. Next, take a new, clean slide, place it on the surface of the first slide, and draw it back into the drop. Then, push the spreader slide to the other end, and dispose of the spreader slide in the red biohazard bag. When performed properly, your slide will appear similar to this. Finally, allow your slide to air dry and then observe underneath the microscope. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.